let's get started. This is too loud, as usual. Okay, so I just want to say a few words about the exam that will start Monday and end Wednesday. So it's, as you know, it's going to be a take-home exam that I'll hand out at the end of lecture on Monday, and then you'll hand back at the beginning of lecture on Wednesday. There will be 10 questions on the exam. 6 of them will be shorter answer, and 4 of them will be longer answer questions. The way we do it is that we grade all of the questions, but in order to get 100, you only need to get sort of 5 out of 6 from the first section and 3 out of 4 from the second section. So whatever your best scores are, those are the ones we count. So the idea is to try all of the questions, and to get a perfect score, you don't have to get all of them right. You can miss part of one and part in each section. And the model for you to think about like what the questions might look like is the study questions I've been giving you. And so on Monday, we'll have a review session, so you'll get to ask me whatever questions you want about the study questions or anything else, and we can go through them in class. Okay, so let's get started on today's lecture unless there are any questions.
Yeah, will you be posting the answers to the study?
Yes. So we're in the unusual situation of sort of having the review session and then starting the exam immediately. Normally what I would do is post them right after the review session.
Right.
So let me think about how I'll actually do that. I think I'll do the same thing. So at least while you're working on the exam, you can see the answers. And the idea is on Monday, you should come with questions about the ones you don't understand as well, and we'll actually go through them in class. Yeah, so even when you're in class, like if it were a regular midterm, I would allow you to use all materials you want except for electronic ones. And so the idea is that the questions are not really meant to test your recall. They're meant to test your ability to think about the questions and assimilate new information. So you can use whatever you want except each other and except experts. So altruism points will temporarily be suspended. You know, you can't just post on your blog what's the answer to question number 6, that kind of thing. Right? Okay, so today's lecture is on the universal tree of life, which we'll see what that means in a second, and ecological genomics, and you'll see what that means in a second. And the 2 questions we're going to talk about today are these. What can genes tell us about the history of life? And what can genes tell us about ecosystems? And what's interesting about these 2 questions is that historically they have not been questions that you would consider part of molecular biology. Okay? In fact, in the middle of the last century, there were battles going on within biology departments at major universities between the molecular biologists who were doing the experiments to try and figure out the genetic code and try and figure out how genes worked, and what were then sort of the more traditional biologists who were studying ecology, studying evolutionary biology, studying anatomy. And there were genuine, serious disagreements between those 2 camps. You can think of it as any situation where sort of the modern barbarians are at the gates, you know, and the old timers are sort of trying to protect their turf. That's basically the scenario you should imagine. And so the fact that today we can talk about genomics and the study of DNA contributing to these 2 questions is quite extraordinary. And it represents sort of a coming together again of these 2 disciplines that used to be separate. So in order to talk about sort of how DNA can help us figure out history, we have to start with sort of what that history is and how people have historically thought about studying history in the context of biology. And so one of the sort of classic canonical representations of the history of life is as a tree. And for a long time, biologists have tried to classify living things according to their relationships as they understood them. And so this is an old depiction of that from 1879 from Ernst Haeckel, who's a famous biologist of the 1800s. And at that time, you know, this was sort of an advance, but it was also sort of reflecting some of the biases of the age. For example, the idea that man is at the pinnacle of this evolutionary tree, right? That there are these relationships between organisms, and you have closely related branches, representing organisms that are closely related. So you can see that man is next to gorilla, orangutan, gibbon, chimpanzee. So these relationships are very similar to what we consider today as the true relationships between these organisms. But the idea that there's some kind of progress towards man being at the top, as you'll see, is an idea that's sort of fallen by the wayside for good reason. The idea of representing the history of life as a tree and putting together as close branches organisms that are most closely related goes back to Darwin, okay? And so this is a sketch from Darwin's notebook in 1837. So this is obviously many years ago, and it's probably the most important example of somebody sketching in their notebook an essential idea, okay? And you can see how he sketched. He wrote, "I think," so here's my idea, "I think," right? And then he drew this tree. And what he was representing by this tree was relationships between organisms. And as you know, The Origin of Species, which is Darwin's book that came out in 1859, that was the product of his thinking, was a very controversial book. Darwin took a lot of heat for it. He continues to take a lot of heat for it, unfortunately. And the most radical idea in that book was not natural selection. The most radical idea in that book was that all organisms are related by common ancestry. That is, in particular, that we evolved from apes, okay? And a common sort of way of caricaturing Darwin at the time, and still, is to draw sort of Darwin's bearded face on the body of a monkey, right? And so this idea that all organisms are related is a crucial idea in biology. And one of the indications of it, you already know, which is that every single organism living on this planet uses the language of nucleic acids as its source of information, DNA or RNA. And the genetic code, for all intents and purposes, is identical in every single organism. And so that's a really strong indication that every organism alive today is related by ancestry, that we don't have independent inventions of the same thing in different organisms. And so the notion of the tree is that as time progresses, organisms split, what Darwin would call forming species, right, speciation, and split again. So when you have a representation like this of a tree, what that means is that these organisms are related to one another closely because they share a common ancestor at some recent time in the past. And these ones are also related, but their common ancestor traces back further in the past, and you can trace back as far as you want to find the point where different lineages all have a common ancestor. But the idea is that there's a single root to this tree, okay? That is one source of all living things. And I want to explain this concept of an evolutionary tree more, and I want to tell you about how we actually decipher evolutionary relationships using biological information. But before I do that, I first want to clear up what's usually a source of confusion for people when thinking about an evolutionary tree. And that is that it's different from a family tree. It's obviously related to a family tree because the way we get evolutionary trees is parents have offspring, and offspring have offspring, and offspring have offspring, and so on and so on. But an evolutionary tree has sort of different character than a family tree, so I want to clear up what the differences are. Does anyone recognize this picture? Who's the artist? Frida Kahlo, okay? And so what she was depicting here was her family tree, right? So her parents and her grandparents, right? And I think everyone in the room is familiar with a family tree, okay? Or if it were an organism other than a human, we'd usually call it a pedigree, okay? The idea that you can represent relationships between people who have offspring together, and then people who have offspring together, and then finally children at the end. Okay, and the main property of diagrams like this is that every individual has 2 parents, and 4 grandparents, and 8 great-grandparents, and 16 great-great-grandparents, right? And so this gets confusing when you start to think about an evolutionary tree, because what I just said is that we can trace our ancestry back in time to common ancestors. Okay? So we can trace the common ancestor of all humans back to an individual that lived, 100,000 years ago. Now, that's confusing because it looks like from a picture like this that your ancestors keep multiplying as you go back in time, right? That this individual has 2 most direct ancestors, 4 indirect ancestors, 8, 16, and so on. So how is it that everyone in this room can trace back to a single ancestor? And the answer to that question is that most lineages do not survive. Okay? So Frida Kahlo is actually a good example because when she was age 18, she was in a bus accident, and that left her unable to bear children. Okay? So basically, this pedigree ends here, at least in terms of having children. Right? And so I want to illustrate this a little bit more sort of specifically using an example of a sort of cartoon pedigree. So when we draw pedigrees, and this is done a lot in human genetics and also for like animal breeding and so on, we usually draw males as squares and females as circles. And if a male and a female are producing progeny together, then they have a horizontal line between them. Okay? And then if they have children, you draw a line down from that horizontal line and show the children. Okay? So what I'm showing here is basically 5 mating pairs, 5 couples, right? And what I want to do is consider the fate of a particular piece of DNA in these individuals. So the piece of DNA I want to consider is the Y chromosome. As you know, men have an X chromosome and a Y chromosome, and women have just 2 X chromosomes. So the Y chromosome is passed from father to son and only from father to son. Okay? So what we can do is we can say, well, this guy is going to be a father, this guy is going to be a father, 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 and they each have a different Y chromosome. Okay? And that's what I've colored blue, red, green, NYU purple, and orange. Okay, so now we can ask, well, what happens when these individuals have children? And as you know, in a normal population of people, not everyone has the same number of children, right? So if there are 5 children produced by these 5 couples, it's not like each couple is going to have 1 child. So let's imagine that what really happens is that couple has 3 sons, that couple has 1 son, and that couple has 1 son. Okay? So you'll notice what's happened already, which is that this couple and this couple didn't have any sons, right? So in the course of 1 generation, we've gone from having 5 different Y chromosomes to 3 different Y chromosomes. And then those individuals find partners to have children with, and then they have a certain number of children. So let's say that couple has 1 child, that couple has 2 children, sons, that couple has 1 son, and that couple has 1 son. Okay? So now we've gone 1 more generation, and instead of having 3 different Y chromosomes, we only have 2 Y chromosomes represented. Everyone following this? Okay? And then we repeat again. That couple has 2 sons, that couple has 2 sons, and that couple has 1 son. All right? And now we've gone to a situation where just in the course of 3 generations, we started with 5 different Y chromosomes, and now we only have 1 type of Y chromosome. Okay? And so the way to look at this is to say that all of these sons here trace their ancestry, at least of their Y chromosome, back to that male. Okay? He's basically he is the great-grandfather of all of these males. So all of these males have his Y chromosome, and these other Y chromosomes are gone. Okay? Now, and this will repeat again and keep going forever, but the important thing to realize is that if you look at, say, this male right here, he has 2 parents, right? And those each have 2 parents, so he has 4 grandparents, and he has 8 grandparents, okay? So the family tree is there. The family tree doesn't change. But the evolutionary tree is sort of running the opposite direction. You go back in time and you coalesce at a single ancestor. Okay? And this is a really important idea because that's what allows us to look at say, Y chromosomes existing now and trace them back, right? So for example, if this was now, and our sample in a study had these 5 males, we could say that these 4 males are more closely related to each other than any of them is to this male. Okay? And the way we would know that is we would look at their Y chromosomes and we would see that they have slightly different DNA sequences. And we'll get to, in a little bit, specifically how you actually do that, how you can sort of build an evolutionary tree just by looking at DNA sequences. Okay? Any questions about that? All right. So I've only shown this for 3 generations, but we now know that if we take all of the males, say, in a random sample of human beings, so let's say all the males in this room, and we sequence their Y chromosomes, and we figure out how they're all related, we can trace the ancestry of every single male in this room to a single male, a single Y chromosome of a male that was living in Africa about 60,000 years ago. Okay? We can do that with very high confidence. The date might be, give or take thousands of years, but approximately 60,000 years ago. Okay? So it's this logic, this idea that the more similar your DNA sequence is, the more recent common ancestry you have, is what allows us to build evolutionary trees. So here's an example of an actual evolutionary tree in sort of a modern sense, not in sort of Ernst Haeckel's where you actually draw the tree kind of thing. And this shows the relationships of primates, okay, so monkeys and apes, right? And what you can see is here we are, humans, and we're most closely related to chimpanzees, then to gorillas, then to orangutans. And this has a scale of millions of years, okay? And so, for example, we know that humans and chimpanzees shared a common ancestor about 5 million years ago. Okay, that means a single ancestor that was probably something that looked chimpanzee-like, gave rise to every single human being and every single chimpanzee. You go further out in time, gorillas about 8 million years, orangutans about 15 million years, and you can figure out all of the relationships, and so all primates share a common ancestor somewhere 50 to 60 million years ago, okay? And you can keep going. So you can add to this tree all mammals, right? You can add dogs and cats and dolphins and whales, right? And those will all sort of branch off down here, right? Although not too much further into the past, actually. There was what's called the mammalian radiation that took place where it seemingly all of the different mammal groups sort of came into existence and diversified in a very short amount of time. So somewhere down here, you can imagine dogs and whales and hippos and everything branching off. And you can go even further. So you can go to all vertebrates, and that would include reptiles and dinosaurs and chickens and fish. You can go to all animals, right? Further, further, further down in the past, hundreds of millions of years, including insects and crustaceans and every other type of animal. You can go further into the past, and then you would get back to all eukaryotes. You would include plants and fungi, and this is billions of years in the past, right? And then you can go even further until you have what was the common ancestor of every single animal, every single plant, every single fungus, every single bacterium on the planet, okay? Every single living thing a few billion years ago, okay? And so now it's, as you know, it's pretty trivial to get DNA sequences, genetic information from any organism you want. And so what you can do is you can actually figure out that tree of life that includes all living things. And it looks something like this. So these are just sort of organisms that were chosen for convenience and other reasons, like they're studied a lot. And so basically you take a bunch of living organisms and you get some DNA sequence out of them, and you use those DNA sequences to figure out who's most closely related to whom. It's the same operation you would do if you're trying to figure out which males of a human population are most closely related to each other, but you're doing it over a much, much broader evolutionary time distance. Okay? And so as you can see from this tree, nobody is especially privileged on this tree. Okay? You probably can't read what these are, and I'll go back in a later slide and sort of show you how these break down. But one thing I want to point out here is that humans are like over here. Okay? We're not up here. We're not over here. We're not in any privileged position on this tree. Okay? And in fact, the property of a tree is that wherever you have a branch point, left and right are arbitrary, so you can switch those around. So you could redraw this tree, putting humans pretty much anywhere on this fan-shaped thing. And so if you have sort of this ancient progressive view of everything leading up to humans, then you would sort of rig it so that humans were at the top or over here or something. But there's no sort of scientific reason to do that, because we all derive from the same common ancestor way back when, and we all have been around. All of these lineages are exactly the same amount of time. So who's to say who's privileged, right? You know, we're doing okay. We have a very recent origin. You know, modern humans only about 100,000 years ago. Bacteria are doing okay too. So who's to say we're better than them? Okay, so what's interesting about what DNA has done to the study of the history of life is that in very recent time, so in my lifetime, not in your lifetime, a completely new branch of this tree of life was discovered. So as I've told you before, one major division of all living things right now is between what are called prokaryotes and eukaryotes. So eukaryotes are things like us, things that have cells with a real nucleus in them, with chromosomes in the nucleus, with membranes that the ribosomes are sitting on, and a few organelles inside the cell that we'll talk about in a little bit. So these are things like us, plants, animals, fungi. And then there are prokaryotes, which are basically bacteria. Okay, single-celled things, don't have a nucleus. They have DNA, but it's usually like a circular chromosome like E. coli's, like we talked about last time. Okay, and it turns out that you know, this was the way life was broken down for many, many years until 1977, when it was discovered that there's a whole other branch of life. And this other branch of life is what's now called Archaea. Okay, and these are some of the weirdos that I introduced you to last time. Okay, so there are three major categories of Archaea. They're called halophiles, methanogens, and thermophiles. Okay, and I just want to go through this to sort of give you an idea of how weird they are and why it probably took a long time to figure out that they existed and that they are a different branch of life. So halophiles are things that like salt. Okay, and so this is Owens Lake in California, and lake is a generous term for this time of year, right? So at this time of year, that lake is just a dry lake bed, basically, but it has a thin film of brine on the surface of it. Okay, very, very salty, basically saturated with salt. And if you threw a human being on that and said live, you know, they'd dry up pretty quickly. If you threw an E. coli cell onto that dry lake bed and said, go ahead, try your best, they would die pretty quickly. But there are organisms that love that environment, that basically can live on a very, very concentrated brine solution. It turns out that they're microorganisms, so they're single-celled like bacteria, and they're pink, so that's why this picture is pink. And you can see that they've taken over the entire lake bed. Methanogens I introduced you to last time, they live at these deep thermal vents in the ocean. So this is in the Gulf of California. You can see the depth is 2,000 meters. So over a mile deep, bottom of the ocean, where these sort of sulfurous flumes are coming up, right? And down here, there are bacteria that are methanogens, that oxidize, that don't breathe oxygen, they breathe sulfur. Again, really weird creatures, and you can imagine it would take a while to discover them, because it was only recently that we developed machinery to actually go down to the very deep ocean, grab samples, and come back. Okay? Another really interesting group of these Archaea are what are called thermophiles. They live in really extreme temperatures. So this is Yellowstone National Park. It looks like some, you know, crazy hallucinogen-induced painting, right? But this is actually an aerial view of a real place. Okay, this line here, just to orient you, is a path, and there are people walking on it. Okay, so that's the scale of what you're looking at, right? And this is a sort of boiling cauldron of sort of natural hot springs. And around this area are organisms of various colors that like that environment, that like really high temperatures and really weird concentrations of metals that happen to be here. Okay, so again, sort of a strange environment, strange organism, only discovered relatively recently, and their position on the tree of life was only discovered relatively recently. So the question is, you know, these archaea, they're single-celled organisms, so in that way they sort of look like bacteria, but there were other guesses that maybe instead of being more closely related to bacteria, they're actually more closely related to eukaryotes, okay? And so that's a scientific question, and we can use DNA evidence to try and answer it. And I'm going to show you the answer first, and then we'll go through how that answer is actually gained, okay? So we now have a really good notion of what the tree of life looks like, including archaea. And it looks like this. And so this and a bunch of slides in this lecture are from this required reading article from Scientific American. And so we have these three major branches of life: the bacteria, the archaea, and the eukaryotes. And it turns out that archaea are actually more closely related to us than they are to bacteria. Okay? So, and you can see that the way you read that is you say, okay, this is animal, so we're up here somewhere on one of these branches. And we trace back in time, and we meet the branch that leads to archaea first before we meet the branch that leads to bacteria. Okay? Another way of saying it is going from the bottom, this last universal common ancestor, so the single cell that gave rise to all of life on Earth many billions of years ago. Okay? The first branch is a branch that goes to the left to bacteria and to the right to both archaea and eukaryotes. So that says archaea are more closely related to eukaryotes. That's how you read that. Okay? And so just to point out some major features of the tree, we have animals more closely related to fungi than we are to plants. It was previously thought that fungi and plants were more closely related because they're both vegetables, basically. No more intelligent reason than that. Turns out animals and fungi are more closely related than they are, either one is to plants. Plants and algae obviously are related to one another. And then these are some weird eukaryotes, the kind that you see in pond water, like ciliates like paramecium, amoebas, things like that. Bacteria are over here. So E. coli is a proteobacteria, so E. coli would be found on one of these branches. I showed you a cyanobacterium last time. Cyanobacteria are green, they photosynthesize, and then many other different types of bacteria. And then you have these branches of archaea, and so on these branches are these halophiles and methanogens and thermophiles. And what you can see is that there's one other special feature of this tree, which is that it's not just sort of branching and branching and branching, but some of these branches actually sort of curve and connect again back to another branch. And there are two major events that we'll talk about today, where a eukaryotic ancestral cell ate a bacterium and gave rise to a major organelle that's basically essential to our cells now. So the first event was an ancestral eukaryotic cell eating a proteobacterium, so something similar to E. coli, and that gave rise to our mitochondria. And I'll talk more about this in a second. And then later on, only in the lineage leading to plants and algae, another eating event happened where an ancestral cell ate a bacterium that was a cyanobacterium, and that gave rise to chloroplasts. Okay? And so those two events are what are called endosymbiosis, endo for inside and symbiosis for sort of shared mutual relationship. And so the endosymbiont hypothesis then is the idea that these events happen, that you had a primitive eukaryotic cell which somehow evolved from a prokaryotic cell, a bacterium. And the features of that primitive eukaryotic cell is it had a genuine nucleus with chromosomes inside instead of just DNA floating free. It has these internal membranes where the ribosomes are. And it has what's called a cytoskeleton that gives cells their different shapes. And that ancestral eukaryotic cell at some point must have eaten a type of protobacterium. You can see it's called bacterial food here. So it actually ingested that cell. And normally what would happen is the cell would go inside and it would get digested. The different parts would be broken down and used as food or energy or building blocks for that eukaryotic cell. But in this one case, what happened is the cell remained intact, and a symbiosis developed where what was good for the symbiont, the bacterium, was also good for the eukaryotic cell holding it, probably because the symbiont was then protected from the outside world, and the eukaryotic cell gained energy that the symbiont was producing. And so in an evolutionary sense, they had sort of a gentleman's agreement to sort of stick together and not cause each other trouble. So what you had then was the first eukaryotic cell with a mitochondria in it. And what happened over time is that this symbiont, instead of being recognizable as a bacterium that you could then sort of take back out of the cell and grow, really became more and more specialized as this energy-producing organelle inside our cells. Okay, and that's what a mitochondrion is. And in that process, what happens is, remember, the bacterium has its own DNA to start with, and our mitochondria still have their own DNA. Okay? But they've lost a lot of their DNA. They've basically only kept the things that are required to do this energy production. They've lost all the other things that a free-living cell would normally have. And so you get still, you can isolate DNA that just comes from the mitochondrion, okay? But it's a very small genome. Okay, most animal genomes are only between 10 and 20,000 DNA letters instead of 3 billion letters like in the rest of our genome. Okay, so that was one endosymbiotic event. And then the later endosymbiotic event that only happened in the lineage leading to plants and algae is that a cyanobacterium was eaten in the same way. Again, this agreement was set up in an evolutionary sense where that cell got to stay inside the eukaryotic cell. And that became the chloroplast. And the same thing is true. Chloroplasts have their own DNA, but they've lost a lot of genes. They only retain genes that are used for photosynthesis. Okay, and this notion has really, at first it was a little bit controversial, but it's now a very well-established concept in biology that these endosymbiotic events happen. And the other thing that's happened is this idea of endosymbiosis has entered the popular culture. Do you have a question?
Yeah, it produces its cell wall with plants that came from that chloroplast. Does it gain it back somehow? Same process?
Yeah, so it's sort of a technical answer to your question, which is that there's a cell membrane and then lots of bacteria have a cell wall on top of that. So the cell membrane, in a sense, is still intact, but the cell wall is what's lost. So they still have their integrity as a sort of membrane-bound unit. Okay. So as I was saying, the endosymbiont hypothesis is now sort of firm science, but it's also entered the popular culture. So anyone who's watched like the Star Wars movies knows that Jedi are very high in what are called midichlorians. Okay, so this is like a huge ripoff of mitochondria and chloroplasts, and the idea is that they have these special organelles and that they're at very high levels in Jedi, and that those organelles don't produce energy, they don't photosynthesize, but they allow the Jedi to communicate with the Force. Here's another example that I kind of like about the endosymbiont hypothesis, and that's the idea that the mitochondria are really just hanging out until they get their day to have revenge on us. And so there's a video game called Parasite Eve. Okay, it takes place in New York, right? And here's how this game is set up, right? So here's the story of Parasite Eve. Mitochondria, a microorganism that lives within human cells and has existed symbiotically with the nucleus for hundreds of millions of years. Critical for production of energy within living things, mitochondria has its own genetic code and the ability to evolve and multiply. What if this organism had her own intentions and was waiting for the day to take over the human race? Okay, and so the story starts on Christmas Eve in Manhattan. Aya and her boyfriend go to Carnegie Hall to watch an opera, and suddenly the actors and audience burst into flames during the performance. While the entire theater is in frenzy, Aya is the only one who doesn't combust. Through her screams of terror, one actress laughs loudly by herself. "Who are you?" Aya asks. "I am Eve." Aya takes her gun and moves towards the stage. So begins the final battle for survival between the human race and the mitochondria. Okay, so there you have it. Now you know that it's a solid idea when it becomes a video game. So that's our tree of life, right? Mitochondria, a bacteria that became a permanent resident inside our cells. Chloroplast, a bacterium that became a permanent resident inside plant cells, okay? And basically, what this picture is in sort of cartoon version, this picture is in sort of specific details, okay? And so we have lots of representative bacteria, eukaryotes, and archaea now. And as I said, the way you get this tree, this actual tree based on data, is from DNA sequences. So that's what I want to talk about next. This diagram again shows sort of the bias in what sequencing projects have been done already. Okay, so these are all fully sequenced genomes now. All of these purple ones are bacteria, the pink ones are eukaryotes, and the green ones are archaea. And on this diagram, you can see that the archaea form this tight group, and the eukaryotes form this tight group, and those two branched closer to each other than either one does to this other big branch, which is the bacteria. Okay? And so the other thing that's discussed in this Scientific American article is really sort of the first evidence that came out that these relationships were true. So the first studies that were done involved about 600 species. So this is the actual tree of species that was looked at in the original studies. So again, you see the major branches. You see eukaryotes, archaea, bacteria, all forming these sort of tight groups. You can see the representatives of each group that were used. So we have humans as a representative animal, for example, corn as a representative plant, nematodes are representative invertebrate worms. Then you have other weird eukaryotes like trypanosomes, which cause a lot of diseases. You have the archaea, including these methane producers and sulfur lovers and so on. And then you have bacteria, including, as you'll notice, mitochondria and chloroplasts. So this, in a sense, is the evidence that mitochondria and chloroplasts started out as bacteria, because when you sequence their DNA, it shows up within the bacteria. It doesn't show up within the eukaryotes, even though those are components of eukaryotic cells. Okay? So the way this tree was built was using not the entire genome sequence, but a particular gene. And the particular gene is one that encodes a component of the ribosome. So we talked in a previous lecture about how most genes encode proteins, but some genes encode RNAs that have a function, for example, transfer RNAs. Another type of RNA that has a function is what's called an rRNA, or ribosomal RNA. Okay? And the reason this gene was used is that all organisms have ribosomes. Okay? And so in order to build the whole tree of life, you need a gene that every organism has. Okay? You can't use, for example, the lactose permease gene because not every organism has that gene. You can't use a lot of genes that we have because many organisms don't have those genes. So you have to find a gene that's present in all organisms. And so the ribosome is a good place to look because the ribosome is present in all organisms. Okay? And so there are many RNAs that actually, or there are two major RNAs that contribute to the ribosome. There's what's called the small subunit and the large subunit. So SSURRRNA is an acronym for small subunit ribosomal RNA. It looks like this. Don't worry about what this picture is, right? But you can see that there are letters here. There are A's, C's, G's, and U's. And basically, it's this complicated molecule like a transfer RNA that sort of pairs on itself and gains this three-dimensional structure. Okay? That's not really important to what we're discussing today. What's important is that it's present in all organisms. All organisms have this as part of their ribosome. And therefore, it's good for comparing distantly related organisms because you can actually find the sequence in all organisms. So what was done was to take about 600 species representing all of these different groups and get the sequence of their small subunit ribosomal RNA. Okay? And when you do that, you get this tree, okay? Just by saying, well, which two sequences are most similar? I'm going to put those together. Then which two sequences are most similar? I'm going to put those together, okay? And you build up this tree. So now what I want to go through in detail is how that operation actually works. If I give you some DNA sequences, how do you build this tree? Okay, because that's what all of this is based on. All right? So imagine that we're not looking at 600 sequences, but instead we're looking at just 6 sequences. okay? And these are the 6, okay? It looks like an eye chart, right? So someone in the back, cover one eye and read the top line. Basically, what I'm showing is 6 sequences from 6 different organisms. So I've numbered them 1, 2, 3, 4, 5, 6, but they could be sort of a mouse, a human, a plant of some sort, you know, a bacterium of some sort, and so on. And it's the same part of the same gene. And you can see that because, you know, so they all start with A, and if you sort of blur your eyes, they all look like approximately the same sequence. But there are differences, okay? So for example, where this sequence has a C, this one has a G. Where this one has an A, this one has a T, and so on. Okay? So we can use these sequences to figure out how these organisms, 1 through 6, are related to one another. Okay? And the first thing we do is we simplify the problem. And the way we simplify the problem is we find all the positions where it's identical in all 6 organisms. Okay? And the reason we do that is because those positions contain absolutely no information about who's related to whom. Right? So this first position is A in every single organism. And because it's the same in every single organism, it doesn't tell us how these organisms are related to one another. Okay? It doesn't tell us which ones are more closely related than others. Okay? So we can find all of the positions like that. Right? So there's that one. And there's all of these, where every single sequence has the same exact letter. Okay? And then we can eliminate those, right? Because those don't give us any information. So that simplifies things. And now we're left with just these sequences. Okay? And for each of these positions, some organisms have the same letter, and some organisms have a different one. So this have a C, and this have a G. Okay? And that's information. That allows us to start to figure out which ones are more closely related. If all we had were this position, we'd say, well, all the G's go together and all the C's go together. Okay, and we have 2 major branches, but we can't figure out the differences between the C's and the differences between the G's. But we have more data, so we can actually start to figure out all of the relationships here. Okay, and so what I want to do now is get 6 volunteers to actually do this. So who wants to be species 1? Come up here. Species 1. Species 2. Start running up. We got to get to 6. Species 2. 4 more volunteers. 3 more. Just run up here. Okay. One more. One more. Come on. I'm going to have to pick someone. All right, here we go. All right, so now the task is, you guys are actually not really doing that much. You're just placeholders. You guys have to say which 2 of these are closest together. Okay? So look at the sequences. If you can see them better up there, you can do that. And find the 2 that are most similar to each other. Any 2 that are most similar to each other. 3 and 4. Why 3 and 4? Right, so 3 and 4 are exactly the same, except for the 3rd position is either an A or T. So 3 and 4, we infer then, are closely related species, because they only have 1 difference between them. So 3 and 4, you guys have to start making a tree. So hold your hands like that. Together. Yeah. Okay, so next 2. 2 and 5. Why 2 and 5? Right, so only that second to last position is either a T or a G. Everything else is the same. So 2 and 5 have to go next to each other and hold hands. So you guys move over. Okay, who's next? So how many differences do 1 and 4 have? They have 2. And how many differences do 1 and 3 have? 3. Okay. And so this is the right way to think about it. How many? So now 1 looks similar to 3 and 4. Okay. What about 6? What does 6 look similar to? Where would you put 6 next to? Okay. The other way to think about it is once you've joined these guys and these guys, right, they become a unit, basically, right? So you're not going to then join to 4 or 3. You're going to join like that, okay? So where would you put 6? With 2 and 5, right? Because 2 and 5 have this GCT, blah, 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 and so does 6, right? So if you look at the actual number of differences, 6 belongs with 2 and 5. So the way you do this, right, is, so these guys are a branch, and you have to be sort of a lower branch, right? So you get to join and use your elbow, and then your hand goes there, right? Okay, and 1 does the same thing over here, right? And then if you had the last common ancestor, that would go like here, like this, right? Okay? Okay, thank you. You didn't know what you were volunteering for. Okay, so our tree looks like this, right? So 2 and 5, we're holding hands, and then to 6. 3 and 4, we're holding hands, and then to 1, and then they're all sort of connected down there. That's how you make one of these evolutionary trees. It's not any more complicated than that, okay? The only thing that makes it more complicated is when you have 600 sequences to look at, and they're not 10 bases long, they're hundreds of bases long. And so to do this in reality, you use a computer, right? Because the computer is really good at finding which ones are similar and putting them together, which ones are next similar and putting them together, and so on, okay? So once you have the sequences, building a tree like this is pretty trivial, okay? You get that tree, and then you can start learning interesting things. So this is what the tree looks like if you use that ribosomal RNA gene, okay? But if you use other genes, what's interesting is you don't get the same exact picture. Okay, so here's an example that also comes from this article. And this is the gene for HMG-CoA reductase. Don't worry about what that is. It's an enzyme that's part of what organisms use to make cholesterol, okay? And so if you do the same thing you did for the ribosomal RNA, but you use this gene instead, you get most of the relationships you would expect to get. So you get eukaryotes, you get plants, fungi, animals all related to each other over here. You get archaea, which are related to each other and then related to eukaryotes. And you get bacteria, which are more distantly related to both of these, okay? But then you get one strange positioning, okay? I introduced you to this organism last time. It's Archaea fulgidus. And what I told you last time is it's a methanogen, okay? And it's an Archaean, okay? And so we know that, but why is it ending up over here, okay? Why is its sequence so similar to a bacterium and very different from Archaebacteria, or Archaea? And the answer is that a lot of organisms, especially microorganisms, are promiscuous with their DNA, okay? They sort of let their DNA out into the environment, and other organisms take up their DNA, okay? And so what you get sometimes is transfers of genes that aren't happening from parent to offspring, right? We usually think of the way genes are transmitted is 2 parents have a child, the genes go to the child, okay? You don't think about those parents sort of leaving their genes somewhere and then somebody else picking them up, okay? But that's actually what happens fairly often in the microbial world, okay? And actually, if you think about the experiment you did in lab last week that you're finishing this week, you left some DNA around and some bacteria picked it up. That's basically what you did, right? And so bacteria in general are very good at that. And so what occasionally happens is a gene from one species gets transferred to a very different species. And then that species just keeps it. And so in this case, what we infer from this picture is not that Archaea fulgidus is actually a bacterium. There's plenty of evidence that says it's not a bacterium, it's actually an Archaean. But what we infer is that its HMG-CoA reductase came in the recent past from a bacterium. Okay? We call this a horizontal transfer. Okay? If you think about parents having children, we usually represent that vertically. Parents having children, so that's vertical transmission. Horizontal transmission is not between parents and offspring, but between 2 individuals living at the same time. Okay? And so this kind of event where you find an Archaean gene, a bacterial gene inside an Archaean is called a horizontal transfer event. And it turns out that these are not uncommon. They've happened a bunch of times. And so that leads to a different picture of what the tree of life looks like, more complicated than the first one I showed you, okay? Where it actually looks more like this, right? And so the difference between this picture and the one I showed you previously is that there are all these crossings now, okay? The two main crossings still exist. These are whole organisms that got swallowed up, the mitochondria and the chloroplast. But then you see all these other places where sort of transfers take place, and not just between closely related organisms, but from, say, a protobacterium into an archaean, or vice versa, okay? And so the idea here is that maybe we don't have, if you look across all genes, maybe we don't have a tree so much as like a web, okay? Now, there are some exceptions, right? So as I said, bacteria and archaea tend to be fairly promiscuous with their DNA, but animals in general don't leave their DNA lying around and don't pick up other DNA, okay? And so you can see that this part of the tree, animals, fungi, plants, algae, that's pretty much intact, okay? There's not a web over there. There's not this sort of transferring back and forth of genes, although there is a tiny bit of it. Over here is where you see most of the action. And that leads to the interesting possibility that way, way, way back when, towards the origin of all life, it wasn't a tree, but it was a web, okay? And so speaking of this sort of last common ancestor of all life is a little bit inaccurate then, because instead what you had was probably a lot of sort of swapping around at the origins of life, and you had an original lineage that came into being. Probably life only originated once, okay? But then once you had different types of organisms, they were swapping their DNA back and forth. And so at this very early time in the history of life, a few billion years ago, there was probably not really sort of lineages the way we think of animals, fungi, and plants, but a lot of swapping, and therefore an inability to actually draw a tree at the base here, okay? That doesn't mean that there were, 7 origins of life. It just means that the picture way back here is complicated, and it took a while for it to settle out so that most of the genes in this lineage are actually archaeal, most of the genes in this lineage are actually bacterial, and most of the genes, in fact, almost all of the genes here are animal genes, fungal genes, and plant genes. Okay? And you can read more about that in the article, the Scientific American article. But that's basically the history of life on the planet, okay? Starting out with a common ancestor, a lot of transfer probably in the early stages, eventually the sort of 3 major lineages coming out, Archaea, bacteria, eukaryotes, still some transfer back and forth here, not much transfer back and forth anymore here, and we now have these major groupings of eukaryotes and the major bacteria and archaea. Any questions about that? Okay, what I want to talk about next is the second question that I posed at the beginning, which is, so this is how you use DNA sequences to reconstruct history. And we'll see examples as we go through the course of how this was used to figure out where domesticated crop plants came from, how differences in dog breeds emerged, how humans have populated the planet. It's a very powerful general method for figuring out the history of living things on this planet. The other question that I posed at the beginning was, can we use DNA sequences? How do we use DNA sequences to understand ecology, to figure out things about ecosystems that we would otherwise not understand? Okay? And that goes under the broad topic of what's called ecological genomics. And the motivation for ecological genomics is that, especially when it comes to microbial species, bacterial species, most of them are impossible to study in the laboratory because it's impossible to culture them on a petri dish. Okay? The estimate is that greater than 99% of all bacterial species are impossible to grow in the lab. Okay? Because we don't understand or we can't recreate the conditions that they need to grow. That means that the diversity that we can understand in the lab is only a small fraction of all of the diversity that's out there. Right? And that seriously limits our understanding of natural ecosystems. If you think about different places of sort of species richness out in the world where there are a lot of bacteria, there are a lot of other species, we want to understand those communities. We want to understand how they function. We want to understand how to sustain them. But if we're missing 99 out of every 100 species, then maybe we're missing the ones that are actually most important to sustaining those ecosystems. So how do we study them? We can't bring those into the lab and do anything with them. We don't even know at the beginning that those species exist, because the way you first discover they exist is you get a handful of seawater or you get a handful of soil and you extract, you put it in liquid and you mix it up and you plate it out on a petri dish, right? But if nothing grows there, then you didn't know that there was something living in your sample in the beginning. Okay? So we need some way of studying all of these species that we know are out there but we don't identify individually and that we can't study in the lab by growing them. Okay? And as is a theme in this course, it's DNA that comes to the rescue. Okay? And this is a very recent development. I'd say it's only about really sort of maybe 5 years old as a field. The idea of using DNA sequences to study organisms that are in environmental samples that otherwise you can't study. And it actually turns out to be really powerful and really quick, okay? And so before I tell you about some examples of this, which are actually kind of cool, I want to remind you about sort of the power of DNA sequences, the information content of DNA sequences that I talked about last time. So one of the powerful things about DNA sequences, right, is that just by looking at them and figuring out what proteins those sequences code for, you can tell what functions a cell is performing, okay? And so we took the example of that mycoplasma bacterium that has this really small genome to figure out what sort of the core functions are, right? That every organism has to perform nucleotide biosynthesis and carbohydrate metabolism, including transporting the carbohydrates into cells, lipid biosynthesis to make their cell membranes, and so on, right? So, and a lot of that information comes from just looking at a DNA sequence and saying, aha, this enzyme is encoded, so I know this pathway is there, right? And that allows us to generate these parts lists, right? And part of what we can do there then is say, well, if certain parts are there, then the organism is probably doing a very specific thing, right? So if I give you the sequence of the E. coli 0157 genome, and you find the genes in it, and you say, okay, I see iron transport, I see these toxins, I see those syringe proteins. I know this is something pathogenic because it has those proteins encoded in its genome, right? So this is something we can do in general. We can just look at DNA sequences and learn something about the organism from which those sequences came. So the first really great example of doing this for basically an environmental sample, just sampling an environment and figuring out what's living there and what those organisms are doing, comes from a study that built a special yacht called the Sorcerer II, whose mission was to sail the seas, collect ocean water, and collect all of the DNA within that ocean water, sequence it, and then figure out things about what organisms were living there, okay? And so you might be able to recognize the guy on the yacht as Craig Venter. So he's the guy who came up with this shotgun sequencing method that competed with the public genome project, right? And so after he got done with that, he needed something else to occupy his time, so he built a yacht, right? And set up this private foundation basically to do this kind of research. So here's what they do. You can see that coming off this yacht is a big tube, okay? And what they do is they just collect seawater and they pump it into containers on the yacht, okay? And so if you go to their website, you can see exactly how this all works. So they have this hose that basically samples from the ocean, and then they have these filter tanks. So everything that comes out of the ocean goes through these filter tanks. And you can imagine that if you're trying to understand microscopic things and you just take seawater, you're going to get, crabs and lobsters and fish and plastic bottles and all these other things. And so you go through these filtering steps to get rid of all the macroscopic stuff that you can understand without going through the trouble of getting its DNA. And so basically, all it amounts to is sending this seawater through a series of filters that, the first step is get rid of the fish and the plastic bottles. The second step is get rid of, sand and sediment. And then the final step is to allow only water to pass through so that whatever's left in the sample gets stuck to this last filter. Okay, and by going through these first filtering steps, really the only thing left to get stuck here are bacteria. Okay, single-celled microorganisms. Okay, so then what you do is you take this last filter from the yacht, you seal it up, you freeze it on dry ice, and then you get a helicopter to come pick it up and bring it back to the lab. Okay, so you basically airlift this filter back to dry land, to the laboratory, and then you have this filter back in the lab, and what you can do is you can chemically treat the filter in ways you can probably imagine now, having gone through your lab exercises, to remove everything but DNA. So you know that there are very simple chemical treatments you can do to get DNA out of something, and so you get DNA out of this filter. Now that DNA comes from every single organism that was attached to the filter. It's not just one pure culture of a particular organism, but any organism that was in that 1,000 gallons of seawater is now attached to that filter. Okay? So then what you do is you treat it as if it were the human genome. Okay? You break it up into small pieces, you clone each of those pieces, and then you get the sequence of each of those pieces. So you get a bunch of many, many, many reads of, you know, A, T, C, C, blah, 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 whatever. That's a piece of DNA that came from one of the organisms that was attached to that filter. Okay, does that make sense? So the first place they did this sampling is a really interesting place called the Sargasso Sea. It's named the Sargasso Sea because its major feature is it has this brown algae called sargassum. It's in the North Atlantic near Bermuda, okay? And it's a really interesting ecosystem. Because of the way the ocean currents work, it's a very, very, very salty part of the ocean. It's thought to be a place that wouldn't support a lot of life. It's known that these algae were there, and it's known that certain sort of large organisms use this as a migration route because they can use the algae as a sort of form of protection. But other than that, it was thought to be pretty devoid of life. So this is a good test case. If you can get some seawater out and find actual DNA from living organisms that you didn't know were there, that would be really cool. So that's what they did. They got 1,000 gallons of seawater from this area. They sent it through the filter. They sent it back to the lab. And then they sequenced it as if it were the human genome. So they sequenced in total 1 billion base pairs of DNA. So on the scale, basically, of the human genome. What did they discover? Now remember, each one of those sequences potentially comes from a different organism. And what you can do is you can say, does this sequence match something in the database? Does it match something else that comes out of this filter? And by doing those comparisons, you can figure out approximately how many organisms were in that sample. Okay?
So are they also going through this process of looking for repeats?
Well, so the repeats tend not to fall into genes, and most of the information in studies like this comes from genes. So they basically just ignore the repeats. They're not trying to assemble a whole genome, right? They're just saying, this piece of DNA comes from some gene from some organism. Let me see what I can learn about it. Okay. So they do that. They compare all the sequences they get to existing databases, and they discovered that there are at least 1,800 different microbial species living in that 1,000 gallons of seawater that were previously unknown. Okay? In that seawater. And even more than that, they got sequences that didn't match any organism in the database already. Okay? So 148 species in their sample were not known from anywhere else on Earth. Okay, so this is just 1,000 gallons from one part of the ocean. You can imagine what would happen if you did this on all of the possible environments on Earth. The amount of discovery of species would just be astronomical. There would be thousands and thousands and potentially millions of species discovered that way that we didn't know existed. Okay? One of the really interesting findings of this study was they found out that there were bacteria that were doing photosynthesis, that is, turning light into chemical energy in the form of sugars, but they weren't using chlorophyll. And what was really cool about this is that about the time that they got their sequences out of this project, a discovery was made by people studying a particular bacterium, And what those people had found is that in that really strange bacterium that they happened to be studying that was photosynthetic, they were not using chlorophyll either, but instead they were using something similar to what we have in our eyes as a pigment. Okay, so in our eyes are pigments called opsins. And so what had been discovered only shortly before this study was done was something called bacteriorhodopsin, which is an eye pigment-like thing that some bacteria use to make energy. Okay, it's as if we could photosynthesize with our retina, basically. Now, what was cool about that is that was just discovered. It was discovered in one bacterium. And it was thought, well, maybe that's just like an oddball case. It's an organism that lab was studying, you know, who cares? You know, one organism photosynthesizes with eye pigments, who cares? In this sample from the Sargasso Sea, 782 new examples of a bacteriorhodopsin were found. Different examples, okay? So that just shows you how quick discovery can happen, right? You go from one example of something really interesting, which is that bacteria are doing photosynthesis in a way that's not like the way plants and algae do it or cyanobacterium do it, from one example to 782 examples in a single 1,000-gallon sample of seawater. So that can really change how we think about that ecosystem in terms of the way energy passes through it, right? How light is trapped into energy and converted into chemical compounds that are then used by the cell.
Given if they only DNA information, how was it possible to find out that the bacteria was photosynthetic?
Right, so the inference is that they have these bacteriorhodopsins, right? And if they have the bacteriorhodopsin, they're probably using it for photosynthesis. I mean, you're right, it's possible that they could have this bacteriorhodopsin and use it for something else, okay? But there's no way of knowing that, right? And especially since they're not culturing the organism, so they're not doing that test. But it's as if someone, you know, if you took a random sample of DNA, including sort of mammalian DNA, but you didn't know it, and you found opsins, right? You would say, there's very likely that something in that sample had eyes, okay? Because that's where opsins are usually found, right? And if that thing had eyes, then it was probably having a sense of vision, right? It's that kind of inference that's going on. So in this case, you see that it has bacteriorhodopsin, which isn't used in eyes, it's used in photosynthesis. Okay, so they've outfitted this yacht, and they started out, as I said, in the Sargasso Sea, and they have all of these stops planned. Okay, not a bad trip if you think about it, right? They're just sort of skirting along the tropics, basically, in this nice yacht. And the idea is that these are places where they've already gathered samples and analyzed them, and then they're continuing on and gathering samples all these places. And the expectation is that they're going to discover this sort of richness of species diversity that was never really appreciated before. Here's another really cool example of this kind of ecological genomics. So sometimes ecological genomics goes under the name of metagenomics, the idea being that it's of more than one species. It's sort of a meta-analysis higher than one species. And this was an example where people were studying what used to be a major metal mine. Okay, so you can imagine the ecological disaster that this place was, right? You have sort of tearing up the land and extracting heavy metals from it, and not doing a very careful job of managing wastewater because there was a creek coming out of the mine that was just allowed to carry off whatever was sort of left aside when the mining happened. And so Iron Mountain is a site that was declared a Superfund site, so a major sort of government-funded toxic cleanup site since 1983. And this creek, Spring Creek, was downstream of the mine, and it's basically, it's not right to think of it as a creek. It's right to think of it as a leaking battery, okay? Because it's so acidic, it has such a high concentration of metals in it that it's basically as if you have battery acid flowing down a creek. Nevertheless, there are microbes that live there, okay? Basically, every environment has something that will live there. And in this creek environment, it was clear that there were microbes living there, but these are not microbes that are easy to grow in the lab because the conditions under which they grow are so unusual, right? You don't have a petri dish with battery acid in it, okay? It just doesn't work. The other thing that was known is that because the bacteria were sort of metabolizing everything coming through the creek, they were actually accelerating the toxic runoff. They were making everything worse by the fact that they were there. So there was great interest in trying to understand these microbes and figure out how maybe to sort of reverse the effect they have or deal with the effect they have. And so the approach that was taken is the same that you could take with 1,000 gallons of seawater. You go to the environment where these things are living, you gather up as much material as you can, you filter it by whatever means are necessary until you get the bacteria out, and then you sequence them and figure out what's living there, okay? And so the first sort of information from that is starting to come out, and the hope is that what you learn is about how these microbes metabolize the metals that are going through. You might discover, for example, how to create a version of those organisms that would still be able to metabolize those metals, but would actually prevent them from running off instead of from running off into downstream. And so this is a place where the potential is to use this kind of genomics technology actually to help clean up a really disastrous site. Another really cool example is in recent years, a new type of bacterial community was discovered on the seafloor. So the seafloor is basically mud, right? And it turned out, I don't know how people figured this out in the first place, but it turns out that if you take that mud and you stick wires into it, it actually creates electricity, right? So I don't know why someone decided to try that, but they did. And the reason it creates electricity is that, again, instead of using oxygen, these guys use something else basically to breathe, right? And they're not breathing oxygen, they're not breathing sulfur. What they're breathing is iron, okay? And so just by the fact that they're sort of using iron in their metabolism, they create an electrical current, right? And so if you stick two wires in, you can harness that current and actually make a battery, right? And so what's unusual about these samples is that attempts have been made to get pure cultures of bacteria out of the mud, and you can culture some of the bacteria. But then if you take those bacteria and you put them in the mud again, in sort of sterile mud, you don't get a battery, right? The battery property, this sort of generation of electrical current, is actually a property of this community of bacteria, not just one bacterial species. So again, this is a prime case where you can say, well, okay, I want to understand a community that's living in this sample. So what do? I wash all the organisms out onto a filter, I get the organisms, I sequence their DNA, and then I figure out what's present there. And then maybe by understanding all of the organisms that are present and the kind of functions they do, how do they metabolize iron? How do different ones do it differently? Then I'll understand how this electrical current is generated. And this obviously is of interest because the potential is there then to sort of hook up the ocean as a major source of energy, right? Or have places, you know, that are artificial ocean beds that can do that instead of taking oil out of the earth.